purpose of calculating the generation time of bacteria, we will consider only reproduction by binary fission, which is by far the most common method. As you can see in the upper green figure, one cell's division produces 2 cells, 2 cells divisions produce 4 cells, and so on, when the number of cells in each generation is expressed as a power of 2. The exponent tells the number of doublings or generations that have occurred. The time required for a cell to divide or double is called the generation time. It varies considerably among organisms and with environmental conditions, such as temperature. If a doubling occurred every 20 minutes, which is the case for E. coli under favorable conditions, after 20 generations, a single initial cell would increase to over 1 million cells. This would require a little less than 7 hours, and in 24 hours, it would be a number trailed by 21 zeros. It is difficult to graph population changes of such numerous magnitudes by using arithmetic numbers. This is why logarithmic scales are generally used to graph bacterial growth, shown in the lower green figure. In the yellow figure on the right, please notice that the arithmetically plotted or solid line does not clearly show the population changes in the early stages of the growth curve at this scale. In fact, the first 10 generations do not even appear to leave the baseline, whereas the logarithmic plot point for the 10th generation, 3.01, is halfway up the graph. Furthermore, another 1 or 2 arithmetic generations graphed to the same scale would greatly increase the height of the graph and take the line off this page. The dashed line in this figure show how these plotting problems can be avoided by graphing the log 10 of the population numbers. The log 10 of the population number is plotted at 5, 10, 15, and 20 generations. However, this advantage is obtained at the cost of distorting our common sense perception of actual situation. We are not accustomed to thinking in logarithmic relationships, but it is necessary for a proper understanding of graphs of microbial populations. When a few bacteria are inoculated into an environment such as the large intestine or a liquid growth media, and the population is counted at intervals, it is possible to plot a bacterial growth curve that shows the growth of cells over time exhibiting in this figure. Please note that the logarithmic growth in the log phase is due to reproduction by binary fission or mitosis. There are 4 basic phases of growth: the lag, log, stationary, and death phases. For a while, the number of cells changes very little because the cells do not immediately reproduce in a new medium. This period of little or no cell division is called the lag phase, and it can last for 1 hour or several days. During this time, however, the cells are not dormant. The microbial population is undergoing a period of intense metabolic activities involving in particular synthesis of enzymes and virus molecules. If the character of lag phase in the growth curve is flat, with little growth happening, then the exponential phase is a period of maximum growth. During the log phase, or the exponential growth phase, the cells begin to divide and enter a period of growth or logarithmic increase. Cellular reproduction is most active during this period, and the generation time reaches a constant minimum. Because the generation time is constant, a logarithmic plot of growth during the log phase is a straight line. Eventually, the growth rate slows. The number of microbial deaths balances the number of new cells, and the population stabilizes. This period of equilibrium is called stationary phase. Exponential growth stops because bacteria approach the carrying capacity, the number of organisms that an environment can support. Carrying capacity is controlled by available nutrients. Accumulation of waste space when a population exceeds the carrying capacity. It will run out of nutrients and space. The number of deaths eventually exceeds the number of new cells formed, and the population enters the death phase, or logarithmic decline phase. This phase continues and the population is diminished to a tiny fraction of the number of cells in the previous phase, or until the population dies out entirely. The closed system called batch cultures, in which nutrients are not renewed nor waste removed. Exponential growth lasts for only a few generations and soon stationary phase is reached. However, it is possible to grow microorganisms in a system with constant environmental conditions maintained through continuing provision of nutrients and the removal of waste. Such a system is called a continuous culture system. Those systems can maintain a microbial population in exponential growth, growing at a known rate and at a constant biomass concentration for extended periods. Continuous culture systems make possible the study of microbial growth and very low nutrient levels. Concentrations close to this present in natural environments. Two major types of continuous culture system commonly are used: chemostats and turbidostats. A chemostat is constructed so that the rate at which a sterile medium is fed into a culture vessel is the same as the rate at which the medium containing microorganisms is removed. The culture media for a chemostat has a limited quantity of an essential nutrient, for example, a vitamin. Because one nutrient is limiting, growth rate is determined by the rate at which sterile medium is fed into the growth chamber. The final cell density depends on the concentration of limiting nutrient. The second type of continuous culture system, the turbidostat, had a photocell that measures the turbidity of the culture in the growth vessel. The flow rate of media through the vessel is automatically regulated to maintain a predetermined turbidity, because turbidity is related to a cell density. The turbidostat maintains a desired cell density. Turbidostats differ from chemostats in several ways. The dilution rate in a turbidostat varies, rather than remaining constant, and a turbidostat's culture medium contains all nutrients in excess. That is, none of the nutrients is limiting. A turbidostat operates best at high dilution rates. A chemostat is most stable and effective at lower dilution rates.